And thank you, Bethany, for helping coordinate this, and to all the work you do for the Vermont Myco Society. My name is Jess Rubin. I'm a restoration ecologist and educator. I work both for the University of Vermont ALE department, as well as facilitate an ecological resilience service called Myco-Evolve, offering earthworks education and research. And tonight, I'm grateful to share about investigating Myco-Phytoremediation's role while restoring degraded riparian forested buffers into multifunctional habitats for socio-ecological resilience. And I just want to name that this presentation is not super myco heavy, just to kind of keep it less technical. You will get a really good overview on just a person's attempt to restore degraded ecosystems and exploring the role of fungi and plants in that. We definitely have some takeaways that this year's data revealed, which I'm looking forward to sharing. Just a seed of this research is really from 2010, when I used to teach at Orchard Valley Waldorf School in East Montpelier. Students were on a camping trip playing in the river, and a couple of days later, I got a panic phone call from one of the parents whose child was in the ACU with kidney failure, and it was pretty scary. Luckily, they're fine today, but we tracked it to E. coli, a strain of E. coli that was connected to cattle upstream. And at that moment, I just realized, there really isn't enough green infrastructure protecting water quality from land management practices, and got really inspired to pack up the homestead once that position was over and moved to get more training in remediation. And so the first project that we did was working with King Stropharia rugosa annulato. You may know is a secondary decomposer. You may know them as wine caps. And what I learned about King Stropharia was that it was able to, we grew these mycelial mats through a small business grant, partly through Myco-Evolve's early days with UVM. And when you poured lagoon water laden with E. coli through these mycelial mats, you could kill E. coli, not only if it went through the mycelial mats, but even if just the enzyme exudates, the enzymes, kind of chemicals that are released from that mycelium, that lagoon water was in contact with them, the E. coli would die. We were pretty excited about that finding, and we're ready to then create a whole series of pilot projects to have mycelial mats between all the dairy lagoons where that was getting into tributaries. And then we just checked on phosphorus and found out that while the King Stropharia helped decrease and kill E. coli populations, it was actually mineralizing phosphorus. The microbes in those mats were basically making phosphorus more bioavailable, which is the last thing that we'd want to do in this watershed. That's pollution swapping. So that research hold, and the research I'm about to share with you began. So tonight I'll just talk with you about objectives, research context, a little bit about the pilot foundation, some methodology, a bit of findings, and some implications. The project that I'll be talking about is multifunctional riparian forested buffers with 4 main objectives. And you're looking at a picture of the first year of a degraded buffer in its restored form. You can see remnants of buckthorn. The 4 objectives were phosphorus mitigation, increasing pollinator habitat, facilitating rematuration, and growing eco-literacy. And I will go into all 4 of these in a moment. So the general context of this research is in the field of ecological restoration, and we're focused on the ecotone of riparian buffers, which are these strips of land between above waterway land practices and waterways. And currently, the practices of riparian buffer management in Vermont primarily involve chemicals, particularly glyphosate, to remove non-native species. These buffers are not really maximizing their water quality protection because it's very rare that they're monitored and maintained. And so what happens is when that phosphorus is uptaken in plants through the microbes and fungi, They return back to the soil and water in the fall. Once you see the leaves turning, the process called senescence begins, and everything starts going back down to the roots into the soil and water. Rarely do people who are designing buffers think about pollinators, both in their palettes. They're often just planting a few trees and shrubs that are deer resistant because they're often not fencing and not necessarily tracking fungal or floral succession. And rarely are people thinking that we are, on unceded territory, the Abenaki, and that offering access and collaboration with them would be a wise form of reconciliation. So we are proposing and demonstrating these multifunctional riparian forest buffers, offering a holistic approach to address not only the environmental, but the sociological problems associated with what we call colonial agriculture. So I'm going to go through the objectives briefly. Don't worry, we'll get to the fungi. So the first objective of phosphorus mitigation is really rooted in water quality, right? We go back 84 years to Aldo Leopold's, noting that already freshwater health was threatened by human terrestrial practices. And fast forward to the second quote, which is, more than a quarter century ago, with a decline of 50%, we have a higher percentage now where freshwater ecosystem health is in decline. The threats remain the same: land use disturbances, altered hydrologies, and non-native species presence. In this particular watershed, I'll be talking about phosphorus and macronutrient for growth. We're going to be focused on the 38 to 41% that's coming from base loading from agriculture, non-point source, meaning that you can't really track where, but it's coming from like larger areas on the land where it collects and enters water bodies. There's direct evidence that the bioavailable form of phosphorus creates these cyanobacteria blooms. If you've tried to go swimming this summer, sometimes you may have seen this sort of green pea soup-like water with signs that say cyanobacteria beach is closed. That's an increasing phenomenon, not just in this watershed, but around the world. The second objective of pollinator habitat is because we're in the 6th grade extinction. And as E.O. Wilson talks about, insects are the foundation of the trophic web. You can see here coldsplit and bloodroot ephemerals from the restored plots, and later in the summer boneset and Joe pieweed. And you can see here a turtle head with a little bumblebee in there. It's known that our pollinators are in serious decline, and we are basically following the strategies of entomologist Doug Tallamy, who has this idea of the homegrown national park, where you introduce hosts of those species and you could slow down and potentially reverse the decline of invertebrates, and thereby slow down the extinction. And so the next objective of rematriation, this is Carol McGranahan, an Abenaki grandmother we worked with, and regardless of the debates going on right now in terms of the Abenaki, it's uncontested that we're on unceded Wabanaki territory. And she wrote this land acknowledgement, really reminding us that this land is called Nandakana, which refers to the large swath of land going all the way to Maine, people of the dawn land of the east, where the sun rises, and paying our respects to the ancestors, not only of current and past, but also future, and being rooted in gratitude in our role as stewards. And really the importance of rematriation is a concept that refers to land back to Indigenous people, particularly the women who tend to be the medicine keepers. And as a woman of white skin, I have the responsibility to do what I can to leverage access in colonial spaces to ask these questions of what would reciprocity look like. And we know from Standing Rock days on that connecting with the land and ways of the Indigenous people and advocating for their sovereignty is directly connected to protecting the life force of the earth. Eco-literacy is a fourth objective because we are in a time, as I've mentioned, the 6th grade extinction. I've often heard this next generation called the janitor generation. I'd like to call them the remediation generation. These are all groups that came out this summer from King Street and EcoCorps and AmeriCorps and Restoration Corps and Myco-Evolve's Homeschool and NOFA. And just the importance to train the next generation, growing that eco-literacy and helping them develop that inherent sense of care with the skills. So in this picture here, you can see two studies. I'm going to talk about the pilot briefly and then go into the expanded study. And this is the lake right here. This right here is Rhamnus cathartica. This is what the buffers look like beforehand. So this whole buffer going all the way down to the lake is pretty much 95% Rhamnus cathartica, common buckthorn. And you can see very limited diversity in the plant understory. And this is a view of restored plots looking up at the compost piles. So there's a compost pile at the top of the hill, which is part of the problem. And the green and red, this is a random block design. So they each represent plots, and the green and red represent plots that we restored. The black represents plots that are the control. They'll be yellow when we look at our data. And the green are the ones that are inoculated with mycorrhizae fungi, which we'll learn about. And the red are not inoculated. And so again, these four objectives, these are just different views of areas of the buffers that were restored. And I just want to mention briefly that, you know, while we don't speak poorly about any plants or fungi in general, because we don't know they may be ecosystem placeholders, this Rhamnus cathartica not only does not provide a lot of understory species, it also doesn't necessarily uptake a lot of phosphorus. We analyzed its above ground biomass of phosphorus, and it's less than native willows and native dogwood. Also, it does have these flowers in early spring that do provide a little bit of food and nectar for some native bees, but it's a limited window compared to what native plants can provide. And as the genus species name indicates, it can create a cathartic kind of emetic, vomiting phenomena among animals and humans that would eat the berries. It's not providing nutrition. It's actually can be used to remove poisons from the body. I will say that in Nordic traditions, it has a very mystical medicine, and it basically came over in the 1800s from some colonial farmers who thought it was a good hedgerow, which it was indeed. And so again, with humility, we remove this species to make space for others, and we're still obviously curious about many things we don't know. And again, this blue-green algae is another phenomenon, not only of colonial agriculture, but also industrialization. I'm going to now go into the historical trajectory of this site, as one does before you restore a site. You want to go back as far as you can. And I will say that it does not seem that there were these algae blooms pre-colonial times, at least based on what we found in the records. So we're going to go all the way back in time to 407 million years ago. This is during the Devonian era, when plants were moving from water to land. If you think about that time period, the soil was very thin. And we found in Rhine-Chert these organelles called arbuscles, which basically your inside plant roots indicate that these fungi were already living inside these very early primitive terrestrial land plants. And it would make sense because fungi had already been on the planet 500 million years previously, so had those mining skills to help feed those plants in these very nutrient poor soils. And I find it interesting that these fungi helped plants move onto land from water, and now we are working with them to potentially protect water from land practices. So I'm just going to break down some of these jargon terms. Myco-Phytoremediation. So mycorrhizae, right? Myco referring to remediation is cleaning up. And rhizae, when we're talking about the type of mycorrhization we're focused on, are these fungi that grow in or around roots, right? That's 80 to 90% of all plants have mycorrhizal partners. Now, there are many other forms of mycorrhization. As I started the presentation with the saprophytes like Kingstropharia, people use oyster mushrooms, right, that can break down petrochemicals because those hydrocarbons, cellulose and lignin, are very similar, right, to petrochemicals. But in this case, we're focused on working with mycorrhizae, and there's 7 types of mycorrhizae. We're only focused on AMF and ECM. These are 2 categories that most commonly are associated with agroforestry, agriculture, unless you were, of course, growing blueberries, then you would have ericoid. Phytoremediation also refers to cleaning up. However, phyto refers to plants. And the many forms of phytoremediation, the form that we are going to be practicing and talking about today is phytoextraction. So basically you're harvesting the plant when it has the maximum toxin, if you will, in its above ground body. And thereby it's no longer in the soil to the concentration it was. When you harvest that plant, you're removing it from the ecosystem. And obviously where it goes next is very important. Put that together and you have this myco-phytoremediation. And just worth noting, the relationship between the mycorrhizae and fungi, right, is a really important kind of stage crew, if you will, of soil structure and ecosystem multifunctionality. And here's just a quick picture, right? So the, metabolism of plants practicing photosynthesis bring down from that carbon dioxide, right, these sugars to expand the root zone, the rhizosphere, that are mining these formerly inaccessible nutrients, feeding that back to the plant. And this relationship, while it's symbiotic, is not, it can be parasitic. So you may have known about this new ancestor of ours, Michael Phillips. One of his last gifts to us was this book, Mycorrhizal Planet. And he talked about phosphorus. What mycorrhizae can do on farms. Phosphorus acquisition is one of the many offerings. In this particular case, 4 to 1,000 times of what plant roots would get on their own. So keep that in mind. I'm going to now jump into the pre-colonial and then colonial history of the site. And we're going to get back to mycorrhizae in a moment. Thinking about these networks. So in pre-colonial times, as you can see from this map, the land was tended in a very different way. First, we have the Abenaki ancestors moving seasonally as the glaciers were retreating. And Vermont and pollen core studies indicate that as the terrestrial succession moved from like fungi, right, primitive land plants into conifers and then angiosperms, there were abundant mycorrhizal networks. The way that the Abenaki tended the land, which we call permaculture today, you would say through polyculture cropping, and they did have some fires, but in general, we're maintaining these networks. Fast forward, now you have states, you have countries, and you have watersheds and sub-watersheds. This is the one that we're focused in. As we go through this trajectory of colonial land management, I invite you to consider a couple things. So one of them is think about what is going on to the underground networks. And two, if you imagine the soil matrix as a movie theater, which each seat being filled with phosphorus, how we've gotten to a situation where the movie theater is full and running into the street, AKA into the waterway. So early 17th century, 90% of Abenaki are killed. And the first thing that the people who came over here did was they cut down a lot of the trees. Now, mycorrhizae are obligate hosts. So and that means that when the trees died, all of these networks would die. Fast forward, you have the sheep craze in Vermont in the 1840s. There were 1.7 million sheep in Vermont. That's a lot of phosphorus entering the soil. Then you have increase of flock and herd size with refrigeration and the railroads. And then we have more fragmentation of the soil networks through roads and ditches, and they weren't necessarily revegetating. And then a lot of the farms, like Shelburne Farms, accepted subsidies from the government at that time to apply technology coming out of World War II, superphosphate fertilizer, which at that time was the best thing since sliced bread in terms of its ability to boost crop production. No one really thought about its effects on water quality. And then, of course, Dutch elm disease took out some of the remaining trees that did host some of those underground networks remaining. A lot of non-native species came in. And I think it is just worth noting that Shelburne Farms is a remarkable endeavor of several families with the help of the Vanderbilt legacy of basically consolidating 300 farms into this 1,400 acre lakeside area of many different ecosystems. And that's what we'll be talking about today, the research occurring there. This is an arbuscle. If you remember that organelle I mentioned was found in Rhine-Shirt. This is from inside a plant root, and it's where a lot of the nutrient exchanges happen between the fungi and the plant. And these are vesicles, again, inside a plant root. These are organelles, like both of these we use when we're counting colonization in our roots. And this is a literature review that I first co-wrote with a colleague of mine when learning about mycorrhizae's ability to move phosphorus out of the soil and water 10 to 100-fold, and wondering, why is no one applying this to water quality in a watershed where phosphorus is, potentially becoming a toxin when too much of it is entering the water. And so we put together this literature review that summed up a lot of mesocosm and field studies. And the skinny was basically saying, yes, it does seem that when you work with mycorrhizae, it decreases phosphorus runoff, and particularly when you apply phytoremediation, and we need a lot more studies. So once we got that out there, we then began to get to work and create a field study. So this is the site. This is Orchard Cove, a hotspot for both phosphorus and E. coli. I will only talk about phosphorus today. Due to the geologic history of being a glacial lake and the Champlain Sea, we had glacial-lacustrine soil, and that area has been in pasture. There are broken tile drains under here. If you're not sure what that is, these networks that basically funnel water out of an area that's too wet to farm. And it's worth noting that when we took water samples where this lower thumbtack is, it was 18 times what Vermont water quality standards say any water leaving a tributary should be in terms of phosphorus concentration. So definitely problematic location. And I just want to mention that we're not pointing fingers at Shelburne Farms. They inherited a phenomenon that many farms, not only in Vermont, but in the Northeast, inherited from the colonial trajectory I shared, having what we call now legacy phosphorus and current inundations. The fungal networks were disturbed or absent. There's a lot of areas in the farm that are out of management, and then native species came in, and there's a lot of systems that farmers can't necessarily change. And oftentimes it requires people like myself who are not on that farm staff to come in and help. So I just want to mention that this is a Sisyphean project, if you're familiar with the Greek myth of Sisyphus pushing a rock up a hill. Because the amount of buckthorn on the farm is gargantuan. And though in our research area, we removed it, there's a lot of vectors of berries, birds, rodents, and wind. It's also worth noting this compost pile is still receiving 550 cubic yards a year. We did create a vegetative treatment system that was approved by UVM Extension and submitted to the farm, but they're not able to move it at this time. So if the faucet is not being turned off, there's no way we would ever be able to effectively remediate. You see our study site is in between what we call the critical source area and the water. So we still have these surface and subsurface inundations of phosphorus. Now, if the pile was removed yesterday, research indicates it would still take 6 to 9 decades because phosphorus is a very slow moving element. It's not like carbon and nitrogen. It doesn't have a robust air phase. So this is a slow game. This is a long game. And because of that, this is more of a demonstration project. And then just mentioning that there's these unmapped and broken tile drains, which can definitely contribute to soil variability because they may be entering different parts of our plots. We're not able to locate them. So I'm just going to jump into the skinny of the pilot before sharing about the new study. We had one of each treatment. Again, the control buckthorn in place, remove the buckthorn, put in native polyculture species. The green represents the inoculated plot. The red is the uninoculated. It's quite beautiful. If you ever come to Shelburne Farms, I recommend checking out the pilot. It's now 7 years old, much taller than me. And some findings we had from that, we basically monitored it. And so it was like a 4-year project. Manual removal of buckthorn 3 times in 2 seasons works. Mycorrhizae seem to reduce the soluble reactive phosphorus concentrations. However, mycorrhizae did not improve plant vitality nor plant uptake. So what that means is the plants in the inoculated compared to the uninoculated did not have more phosphorus in them, nor were they more, robust. However, the biomass of phosphorus in this above ground species did differ between species, not treatment. 1.7 more plants than we planted showed up, which indicates autogenic succession. Elderberries had the highest concentration, 3,000 milligrams per kilogram. So if you do back the envelope calculations, you can basically figure out 0.1 to 0.5 kilograms per hectare per year. But that's definitely underestimating the amount of phosphorus that you could prevent getting in the lake, because that's only the elderberry, and that's only obviously practicing the honorable harvest a quarter. But if we harvested many of the other species that also uptake phosphorus, that math would be a lot higher. And so if you want to geek out on this, we have 3 published papers on this you can look up, and goes into the skinny of that. So what we did was we improved the study. We tripled it. So we actually have 3 of each treatment, because, 1 of each treatment's never good, but it was all we could do at the time with funding. Instead, the original pilot, we used commercial mycorrhizae, which I highly discourage. And instead, we grew native mycorrhizae. The palette was originally only 87% Abenaki friendly. Now it's 100% edible, medicinal, and utilitarian to the Abenaki. And we added that 4th objective of eco-literacy. So again, just looking at it, you can see that this is the new study further up and we have random block design, 3 of each treatment. So I just want to mention that there really is no need for glyphosate to remove Rhamnus cathartica. This can also be applied to the other non-native buckthorn in Vermont, Frangula alnus, and the non-native honeysuckles. You can see that in the buckthorn that we cut, all the beautiful asters and St. John's wort and turtle heads, grew up around it. Here you see some Trametes species showing up on the stumps. So this technique we learned from Mike Ball of Got Weeds. 3 times in 2 seasons, you cut back that buckthorn and you have a 90% death rate, and then you just cut back that last 10%. Meanwhile, you have the stumps for overwintering invertebrates, fungi, early migrants. And research indicates that glyphosate actually can contribute phosphorus to the water table. It basically can bring soil closer to phosphorus saturation. Now, because originally we were going to write a manual for the state, I did half the plots with volunteer labor manually and half the plots with a mini excavator, because we're thinking about larger organizations that might say, well, how are you going to do this for 4 acres? And you can do it certainly with a mini excavator. It had a third of the time, third of the cost, but it was very little educational value. And I still obviously had to get the soil out of the stumps. Covington clay is, not easy to work with. So I would say to people who are doing it large scale, have all your volunteers and staff come for a couple days and get great workouts and provide snacks, and then you can bring in the machinery, but you certainly don't need glyphosate. And actually, the jury's still out. You might not even need to remove the stumps. We kept the stumps by the waterways to not disturb the water and the phosphorus, but away from the waterways is where we pulled them all out. I think you probably could just leave them in and plant all around it, no problem. This is the palette, and you can see the edible, medicinal, utilitarian uses. These are the AMF and ECM, different types of fungi categories. There's a couple like Salix that are both ECM and AMF. These are the blooming times of the flowers, March through November, and these are the hosts. This is just a view of the concept map of how we planted. This is the waterway, and there's a method to the madness of why we planted what where. And then this is just really interesting information about how we grew the endemic mycorrhizae. We went to the most wild part of the farm, grew them out in pot cultures based on a guide we published at SARE you can get for free. I will say that from our data this year, we learned some things. So we're going to make some amendments to that, which I'll share near the end of the presentation. And then real brief, getting into methodology, we pasteurized low phosphorus soil. We potted up 2,200 bare root plants, half of them inoculated, half not. Meanwhile, we cut all the buckthorn in the plots, leaving all the native trees, and then removed the stumps away from the waterway, loosened up these are some of the potting team, and a lot of groups and a lot of people were involved. Loosened up the Covington clay with broad forks and hand tools, leaving again native species in place, laying down the quire, which is now decomposed, but at the time to prevent erosion, planting according to the planting map, putting up a fence, and then of course the water truck keeping everything alive. And the maintenance, besides, bringing everything from sticks at the Hort farm to actual plants and keeping them watered. Also, this is an example of that second grow back on the stumps that you just strip back. And yeah, just the eco-literacy, it's worth showing you some of the faces of the amazing people who help, like Vermont Youth Conservation Corps team, an all-women team led by women of color. Vermont Youth Build, if you're not familiar with, is a group of students who struggle with traditional high school. This was their math and science curriculum, putting up the fences, permators, and many other many other groups of people. So in terms of quantitative data for the phosphorus objectives, we gathered total phosphorus, malic, and SRP, which I'll explain those later. Here's Kat, one of our interns, gathering plant material, me drying it and grinding it. In terms of mycorrhizae, we'd gather the roots of the plants, and then we'd have to autoclave them and put them in a dye where basically the organelles will pop out pink. So you can see here like the vesicles. So these are the slides, and then we would quantify colonization percentage. And then we also used a oak field sampler for gathering soil, breaking up each plot into quadrants and gathering composites, which we'd process for these metrics of soil as well as DNA to determine the fungi in the soil. And then for pollinator habitat, we do these transects parallel to the waterway. This is Ruth, one of our interns. This is elderberry dried and ground. This is the soluble reactive form of phosphorus that we would extract from the soil. And then in terms of Abenaki education, this is tobacco. Carol McGranahan teaching people making elderberry, processing, harvesting elderberries, processing, making elderberry syrup. So I'm just going to get into the data briefly and say that, 300 years of poor land management practices, we're not going to heal in a, 2 or 3 year study. But we did find some interesting trends. So when we're looking at soil phosphorus, I just want to explain the three metrics. Total phosphorus is the big pool of phosphorus. Malic is what becomes bioavailable that season. And WEP SRP is the bioavailable form that actually plants uptake. Now, the y-axis in all three graphs is the concentrations of phosphorus, right? And the x-axis are the 2 dates that we harvested the soil in June and August. And the yellow represents the OIV, the control, right? The buckthorn mostly. The RV is the restored plot, and the RVM is the restored plot with mycorrhizae. And you can see in all three graphs the same pattern. There's statistically significant less phosphorus in the 2 restored plots compared to the control, which you would hope. However, there was no statistical significance between the 2 treatments. So in other words, the mycorrhizae did not make a difference. And you will see a difference in the dates, and that's just typical phosphorus cycling. It would go down for total phosphorus and up for the other 2 metrics just because it's becoming more bioavailable. So that's just biology and phosphorus cycling. So in terms of mycorrhizal data, this is not related to one of the objectives, but we thought it's really important. Assumptions. How inoculated were the inoculated plots? And it turns out that they were statistically significant more than the uninoculated, thank goodness. However, the uninoculated plots had 40%. And that is because we left all the native species in place. And so those networks stayed in place. And we're going to see later that that's really significant. And what's really interesting is that of all the categories of plants, the herbaceous, particularly sweetgrass, is really the only one that showed a big difference between treatments compared to the meadow willow and the red osier dogwood and the elderberry, which is what those other initials you see on the x-axis here are. Okay, so moving on, total plant data. I just want to show you, this is interesting. So this is how much the phosphorus the plants uptook. And again, y-axis is the phosphorus concentrations, the x-axis are all the different plants we harvested. So yarrow and boneset are the highest, which is really interesting. Herbaceous species had the most phosphorus in their above ground body, and the lowest was meadow willow and red osier dogwood, the shrubs. Now, when you actually multiply the phosphorus concentrations times the amount, having a different phenomenon. This is called biomass. And in this, you actually see that the highest are the shrubs and the lowest are the herbaceous species. So what that means is if you're trying to maximize phosphorus removal, you're going to have to grow a lot of herbaceous species to harvest and maximize their uptake. But still, we have quantities now of how much phosphorus. Again, nothing statistically significant between treatments. So the mycorrhizae was not making a difference in the uptake. And then plant species richness, as you would expect, there would be statistically significant more plants in the 2 restored plots compared to the control. But again, no statistical significance between inoculated and uninoculated. However, really interesting to note how many species migrated. Now, migrated we're defining as either left in place, came in the first year, or emerged from the seed bed. And it was really phenomenal to see how many, like these dianthus, these pinks, came in. Now, I just thought it was interesting to look at the plant palette of what they say is going to bloom, which is the dark green. And it really was only accurate to what actually bloomed the months of July and October. In the rest of the month, it either oversold or undersold what actually happened. So the actual blooms is this lighter green, and it's really interesting to break that down into planted versus migrated. And what you end up seeing is how much the migrated species really contribute to that pollinator habitat. And you can see in some places, for some reason my cat is deciding to do her scratching right here. You can see how much these migrated species in June and July and September and October are contributing to the blooms in the pollinator habitat. So really interesting thing. And this is just showing you how many are actually blooming each month. Our goal was to have at least 3 blooming from May through November. The witch hazel did not bloom this year, but hopefully next year we'll have that. So I think this is just a glimpse of some things that are going on below the soil in the mycorrhizal community. And I just want to point out that this is unfortunately only our 2024 DNA data. Our 2025 DNA data got frozen in its analysis during the government shutdown, and only is starting to be analyzed now. So keep in mind that this is last year's data. Things will probably be different, but there's some very important kind of things that we can learn from this. So I'm just going to explain what this is. So in this chart, what you see in this figure here is, first, let's just look at the second and the third lines going horizontal. You see pre and post buckthorn. Now those two, the plant species, did not change at all. They're the controls. One was in May and one was in August. And look at the difference just in the fungal communities. So right there, you can just see when you don't even do anything with the above ground plants or the soil, the fungal communities have their own cycling that differs seasonally. Because the inoculated and the uninoculated plots were tested in August, we're only going to compare with the post. And when you look at that, it's remarkable to see that for some reason the inoculated plot had a lot of dung saprotrophs. And so we're wondering what that's about, and we wonder if the enzyme exudates of the inoculum attracted a lot of potential pollinators that left frass. We did see some nibbling, even though there's a fence, a lot of rodents. So we're not really sure what's going on. It's definitely a mystery. And the other thing that's interesting is to look at the top line, the raw inoculum compared to the inoculated, and you can see that it's really not a direct transfer, and particularly in certain categories. So the endemic mycorrhizae community is very complex. Fungal communities arise seasonally, not a direct transfer of fungi via inoculation, dung saprotrophs and the restored inoculated. Now, if you look at this down here in the bottom left, you see a couple of other interesting phenomena. One is that the raw inoculum in this case really only had what we call generalist mycorrhizal fungi, the Galamus generis. And if you look at the restored inoculated, it really, the transfer happened of others of them, but not equally to what was in the raw. But you compare the uninoculated and the inoculated, and you can really see that a lot of that native mycorrhizae carried over. And so this is a big learning for us, because when we grew the endemic mycorrhizae, we had learned to do that when we used to be working on a commercial farm that's an organic farm in a floodplain that feeds the food bank, and we were growing mycorrhizal banks. So what we were using to grow the inoculum was oat straw, red clover, and oats. And we realize now, oh, that was appropriate for that farm, but we really should have grown the inoculum from that forest in all the different seeds of the species we were growing, and that would have brought out a lot more of the fungi. that are in there. So takeaways: endemic mycorrhizae might not be necessary for riparian forested buffers. There's already enough mycorrhizal fungi in there if you leave native species in. We think this research strategy would apply better to areas that are fields that you're turning into buffers to jumpstart. Cyclical harvest, that phytoextraction, is definitely helpful facilitating phosphorus mitigation and can be incorporated into how riparian buffers are managed. Providing this multi-synusial, right, herbaceous, herb, shrub, and tree level can increase pollinator habitats, and co-designing and coordinating with the original peoples offers a model for involving marginalized groups in a reciprocal way, and there's many eco-literacy opportunities. So I just want to sum up that the data we found can be our next level of work, is to inform best management practices for riparian forested buffers, at least with some of the strategies we found that do work. There's a phosphorus pays program that just had a pilot. It's taking a break right now, but potentially our calculations can be worked into the next iteration of that. This idea of helping with the harvest, reclaiming the commons, particularly with what's going on right now in the federal government, being able to increase food and medicine security in our local communities while helping the farmer mitigate phosphorus. We found that early August is when plants have the maximum phosphorus in their above ground body, which happens to be when many food and medicine species are ready. And there's a lot of opportunities for providing service learning and doing the maintenance. And we are aiming to write a manual for the farm, educational manual that can be used for other farms, as well as fact sheets for NRCS. So there is an opportunity to join our team. We're hiring one intern, ideally a college student, and it's a quick turnaround. So feel free to email me if you want that job announcement in terms of the details. And I just want to mention that we are a large group of people that are involved in this. Thanks to all of my colleagues and mentors at UVM and the students that I've worked with, the Abenaki community, the agriculture community, my advisory team, the funders, starting with SARE back in the early days, all the way to Lake Champlain Basin and Nui-Pick, and of course, Vermont Cooperative Extension and Lintelock Foundation and Abenaki and the UVM Ag Department, COWS for hosting us, and Permatours and PAN for supporting, and of course, the Agency of Ag for supporting us through the advisory group, and Shelburne Farms for hosting, and Youth Build and VYCC for helping with the labor. We do have an iNaturalist project that's documenting the species, and people are welcome to go onto that, and hopefully it'll be a resource in the future. And the project ends in December 26th. And when we take the fences down, we'll be incorporating it into a pollinator education program at Shelburne Farms, but people will be able to harness it for research. And so hopefully others will feel free to access it, or at least just check it out. And we will be hosting a tour or two next spring or summer. So I can share that with Bethany, and it'll definitely be on the Myco-Evolve Facebook page and Instagram sites. So with that, I'm leaving a little bit of time for questions. Thank you.
Awesome. Thank you so much. This was really interesting, fascinating subject that I don't know a ton about. So it was very interesting to get an overview. If you don't mind me asking a very basic question.
Yeah, 100%.
Okay, so phosphorus, you want to remove it, right? Because the presence of phosphorus causes algal blooms, and because there's just generally, because of commercial agriculture, there's too much phosphorus in the soil. Is that correct?
Yeah, and so that is correct. And I guess I'll explain it a little more, which is that, obviously we have phosphorus in our bodies, right? It builds our DNA. It's a very important element, and plants definitely need it, right, in their growth pattern. It's really just when it's in excess. And right. So what's happening is 20 to 40% is leaving these areas on farms. And then, of course, in the urban areas, we have problems with outdated septic and wastewater and lack of green infrastructure. But yes, we're basically trying to prevent excess phosphorus from leaving. And one would argue, when we first did this research, that, hey, if you already have what we call legacy phosphorus in the soil, you don't need to do your huge phosphorus additions every spring. You could just get what's in there through mycorrhizae, and some of the farms we've worked with have stopped adding in the spring. But if you're using mycorrhizae, you don't want to be tilling. And so that's the problem in commercial ag. You can't really, you're ripping up those networks. So that's where it gets tricky. Yeah.
Huh. Okay. And I know you mentioned that one of the objectives is to remove the above ground parts of the plant when they store the most phosphorus, right?
Yes, correct.
What do you do with those plants?
Yeah, that's a great question. And so if you remember, the way we designed it this year was 100% of our palette was edible medicinal utilitarian to the Abenaki. So in this year, the Abenaki harvested elderberry and made syrup. We harvested tobacco and sweetgrass, right, which they used for medicine. We did harvest some of the other plants to analyze for phosphorus and not use for medicine, but they could be. So the yarrow, the bone set could be harvested for medicine. The willow could be used for baskets. And every single plant that we had mentioned, even the coltsfoot. Now, obviously, the coltsfoot flowers are in early spring, so it's not all perfectly timed, but you could certainly harvest the coltsfoot leaves. So the idea was that, anything you plant, if you research it with timing, you could harvest, a quarter of it and basically upcycle it back up the ecosystem and use it for tools, medicine, or food. And it's that much less phosphorus going into the waterway.
Oh, interesting. Is there ever a scenario where you just kind of like wholesale remove it?
Yes. I mean, so when we first were doing the research, the advocates of phytoremediation were willow plantations where and their strategy was more for biomass in terms of, like the wood burning pellets and things. And they were quantifying other aspects of what they were removing. But I think we had an article published in Restoration Ecology, and the title was something like Balancing Objectives. And the reason why we called it that was we did the calculations for both. If you harvested all of it, yes, you would have much higher phosphorus removal. But our other objective, which was pollinator habitat, you would then remove a lot of the overwintering, the stems for overwintering, right, species and early migrants. And so because we work with the Abenaki and they practice often the honorable harvest, which is what like Robin Wall Kimmerer, right, in Breeding Sweetgrass mentions, we only do a quarter of each species, which I still think if you had a drone over the entire lake and you were able to pinpoint every single place that non-point source pollution was coming, and you could do some version of this, and say on Mohawk territory, the other side of the lake, they co-designed the palette, and everyone did, a quarter, you could add that up. That would be a lot of phosphorus being, upcycled. Obviously, if you're in a Superfund site and it's an urban area and it's not just phosphorus, but it's, heavy metals, then it's a whole different ball of wax. You're not going to be, eating or ingesting that. That's a different strategy of what you would do with that plant material.
I guess you would have to put it somewhere, like sequester it, because otherwise it just would break down again and release those nutrients back into wherever it was put, right?
Yeah, so the phytoremediation, I stopped recently working in urban areas, and I might go back to it at some point, but partly because of the complexity of that.
Right.
So people I know who do that work, one of my colleagues who's now Dr. Danielle Stevenson is very well known for doing that work in LA. And basically what she would do with these plants is there's like 3 different modes. One of them is incinerate, and then you just have this very tiny little bit. The other option is this sort of dilution where you have this huge compost pile and it's so big over time that the parts per million are relatively negligible. However, I don't know how many years it would take and how much plant material. And then the third option is there's some people like Paul Stamets was able to demonstrate this. You have to have a lot of money would basically be able to extract that metal out. But that's, you got to have super high technology. But again, I feel like both mycorrhization and phytoremediation are baby fields of science. Bioremediation is much older, right? Even in the Roman era, right? They were using microbes a little bit for some sort of cleanup. But I think that a lot more can be figured out if we can get that national definition changed and more funding can go to train people. So
Makes
it's a young field. There's a lot to learn about it still.
Well, it seems very exciting.
Thank you so much, Bethany.
Thank you. And thanks very much for presenting your lecture. I'm going to stop the recording.